Okay, so let's come back to our scenario. What is protein structure? Can you define that? When we talk about protein structure, we are referring to the arrangement of amino acids in a protein and the extent to which we have folding in that in these chains of these amino acids, and this gives us four different types of protein structures. Okay, so when we talk about protein structure, we are looking at the chains of amino acids that form a particular protein. And these chains can actually fold to give us different shapes of these amino acids. And then the other thing that we can talk about are the interactions or the bonds that we have in these chains. So if you can talk about the things that refers to the protein structure that you're talking about. And we know that we have four of them: primary, secondary, tertiary, and then quaternary. Okay, and we can basically say that with primary, we are talking about sequence of amino acids. With secondary, we are talking about sequence of amino acids and then the folding that we have, beta and alpha. And this, of course, influences this solubility and the function and structure or stability of these proteins. We now introduce in the hydrogen interactions or hydrogen bonds in secondary. And then in tertiary, we are talking about chains of amino acids, or basically polypeptide chains, but these now have more interaction. Van der Waals forces, we have the disulfide interactions, ionic interactions play an important role. And these are usually more stable. Okay, and then when we talk about quaternary, we have different chains now. Okay, and these different chains, again, you have the same kind of interaction that we talked about earlier on. And this is where you have hemoglobin. So hemoglobin is actually a quaternary protein. Okay, if you change this sequence of amino acids, you're changing the structure of the protein. It becomes a different protein. And if it becomes a different protein, it will not perform the same roles. Okay, so that's very important. Even if you only change one amino acid in there. And for example, if you have an alteration in the amino acids of, for example, hemoglobin, you can get a disease. Okay, so sickle cell is actually a disease that happens because one amino acid is being replaced by another. And people who have sickle cell have a lot of problems, health problems that can actually kill them before they reach advanced age. Okay, so that is that. And then the other point is the issue to do with denaturation is that you see these different proteins that we have in our bodies have different structures, like we have looked at them. These structures can be changed because of heat, because of acids. What these heat and acids do is they break down the bonds. Okay, this way that is the peptide bonds, way that is the ionic bonds, way that is the disulfide interactions, and all these other interactions that we talk about. And when this actually happens, they change in appearance. And this is what the students will actually see with the white egg. And then if you have a protein that has more than one chain, one polypeptide chain, that definitely is a quaternary protein. And hemoglobin is an example. Hemoglobin carries oxygen or even carbon dioxide in our bodies.